Hi guys, this is a review video on gas transfer and regulation of breathing for Lecture 31 and 32 of Physio. Let's get started. We have only one equation, and we'll take a look at that in a second, but how to calculate the oxygen content in the blood. We're looking today at, now that we have oxygen and carbon dioxide in our circulatory system, how do we actually get them to where they need to go? So starting off by looking at oxygen. Oxygen is carried in two forms through the circulatory system. The first is dissolved in plasma, and you can see we have our alveoli, oxygen coming across the membrane, just dissolving into the plasma in the blood vessels overall. This is relatively low percentage, and the amount is proportional to the partial pressure of oxygen in the alveoli. And so basically the idea was that for every 1 millimeter of mercury of partial pressure of oxygen, you get 0.003 milliliters of oxygen will dissolve into the plasma. And so for like the normal partial pressures of oxygen we see at atmosphere and in the arterial blood, or in arterial arteries, that only gives us 0.3 milliliters of oxygen, which is not very much oxygen. So we have more than just oxygen dissolved in plasma. We have to use hemoglobin as well. And so the oxygen that goes through the membrane will also be taken up by our hemoglobin. We're familiar with hemoglobin, as we know that one hemoglobin molecule can bind 4 molecules of oxygen. And we have this nice equation for calculating the oxygen content per 100 milliliters of blood, and basically it's hemoglobin concentration times the amount of oxygen 1 gram of fully saturated hemoglobin can support, and then the degree of saturation, which is typically 1 in these equations. All right, and then the hemoglobin concentration is slightly different for men and women. We're given 14 grams per milliliters of blood for women and 16 grams per milliliters of blood for men. So these are static values you just plug it in based on if you're calculating for a woman or man, trying to calculate the O2 content. Milliliters of oxygen per 100 milliliters of blood. All right, so that's not too bad. We have, of course, our oxygen taken up by hemoglobin, and then when you get to the tissue, we want our oxygen to disassociate from the hemoglobin, go back into our plasma, and then dissociate or diffuse into our cells overall. All right, and so before we talk about oxygen dissociation from hemoglobin, this is just another illustration of what's going on when we introduce hemoglobin as an oxygen carrier. So here we have a nice equilibrium state. We had a partial pressure here, and as a result, we had oxygen molecules move on both sides of our membrane until it reaches equilibrium, and then you add a hemoglobin molecule from our red blood cells, and you can see that it has attached 4 different oxygen molecules. All right, and so now you no longer have a nice 5 to 5 equilibrium of oxygen molecules, because we have 4 that are bound here, and so you get more oxygen molecules diffusing across our membrane. Then we have another hemoglobin molecule coming in, picking up even more hemoglobin, or more oxygen, and then you have additional diffusion overall. And so you can see that as a result of hemoglobin, you can get a higher concentration of these oxygen molecules on, on the side of our vessels in comparison to if you just use the dissolving in plasma overall. All right, so then once we're in this situation, we want our hemoglobin to release the oxygen. We have a couple of different factors that can influence how readily hemoglobin disassociates the oxygen molecules. All right, and so this curve right here is just the sort of standard percent saturation curve with partial pressure of oxygen. And so the idea is that as you go towards lower and lower partial pressures of oxygen, you're going to have a decrease in percent saturation, right? And so that's kind of saying something that is maybe fairly obvious. You're going to, as you get to tissues that are further out, you're going to unload your oxygen from your hemoglobin. And the factors that can affect this curve, and so, we have, if you have an increase in temperature, then you're going to have a shift of the curve to the right. And so basically what this is saying is, if this is our usual curve, then increasing the temperature will have the molecules be more excited, and they have more energy, and the oxygen can disassociate from the hemoglobin just due to the fact that they're moving around more, right? And so you see this rightward shift of the curve, and you can see that, just trace it down at the 50% mark, you can see in comparison to the this orange normal curve here that you have rightward shift, but for the same 50% saturation that occurs at a higher partial pressure of oxygen. So basically what it's saying is, if you are increasing your temperature, you have an increase in the concentration of DPG, which is 2,3-disphosphoglycerate, which is a byproduct of, this is from glycolysis. Then you're going to have a rightward shift of your curve, and you will unload oxygen into the tissues at higher partial pressures surrounding the hemoglobin. Right, so you don't need as big a partial pressure difference of oxygen to unload the oxygen into the surrounding tissue. All right, so the same thing here, if you have an increase in carbon dioxide, this is going to give you an increase in concentration of hydrogen ions. All 4 of these are going to serve to decrease the hemoglobin affinity for O2 and allowing for unloading of oxygen at a higher partial pressure of the surrounding tissue. All right, then worth mentioning, so these are all rightward shifts. If you have carbon monoxide, there's a super high affinity for hemoglobin. It will do a leftward shift, actually increasing the affinity of oxygen for hemoglobin, but carbon dioxide will outcompete the oxygen overall. All right, so then we have carbon dioxide transport. So, we want to get our carbon dioxide as a waste product from glycolysis out of the body. So we're kind of going in reverse, but we do have carbon dioxide, just like with oxygen. Some of it is dissolved in the plasma. This is about 6 to 7% of the total carbon dioxide that needs to be removed. Then we have carbon dioxide stored as bicarbonate ions. And so, we have this chemical equation here, carbon dioxide mixing or reacting with water and producing our bicarbonate and our hydrogen ion. And without a catalyst, this is pretty slow, but it does happen. If the carbon dioxide goes into a red blood cell, it will get exposed to carbonic anhydrase, and it will produce this bicarbonate ion relatively fast. And this bicarbonate ion will be pumped out of the red blood cell in exchange for chlorine ions. And as a result, you get a lot of bicarbonate ions in the blood itself. So about 90% of CO2 is bicarbonate in the arteries, and then 70% of the CO2 content in veins is stored as bicarbonate ions. All right, and then the third way that carbon dioxide is transported is through carbeno compounds, which is basically just saying it's carbon dioxide is associating with hemoglobin. It's a relatively small amount 4% in arterial blood, 23% the total content of carbon dioxide in venous blood, and you can see that here we just have carbon dioxide coming in, binding to hemoglobin, forming this carbaminohemoglobin compound. All right, so this is all based on this equation right here. What happens when carbon dioxide mixes with water? You get our carbonic acid and our bicarbonate, but while this will proceed spontaneously, it is a relatively slow process. So you really need this carbonic anhydrase inside of our red blood cells, and then we said that the bicarbonate will leave red blood cells through this chlorine transfer mechanism. Okay, so that's pretty much it. 3 methods of storing carbon dioxide. That was all of lecture 31, so that's not too bad. Here we have some review questions you can take a look at in your, just look at the review document, type out the answers. All right, take a quick pause there. All right, on to lecture 32 material, regulation of breathing. Basically, what we're looking at is the innervation of breathing, and we know from, if you recall from lecture 25 through 27 of physio, we talked about a little bit of involuntary versus voluntary breathing, and recognizing that breathing is mostly involuntary from signals that are starting in the medulla, and then you can also have voluntary breathing, which is typically due to the primary motor cortex of the cerebral cortex, and that will bypass the command center in the medulla and synapse directly in the spinal cord with the intercostal nerves and the phrenic nerves, which innervate the external intercostal muscles and the diaphragm involved in breathing itself. So we have a basis of understanding from previously. We have involuntary from the medulla, then we have voluntary from the motor cortex, but just like with many things, there's more to it. And so we have things like what's going on when you're asleep, what happens if you have emotions, what about pain, startling, what about getting inputs from sensory innervation, what about knowing how much oxygen and carbon dioxide you have in the blood. And so those are going to be the main points we talk about today. But here we have what we're already familiar with. We have our rhythm generator from the medulla, right here. Then those are going to synapse with the motor neurons in the spinal cord, specifically the phrenic nerve, and then we have the intercostal nerves, which are going to our external intercostal muscles and our diaphragm for the muscles of breathing in. All right, we know that our spinal nerves are going to help with forced expiration, breathing out. All right, if we are dealing with these sort of other inputs, emotions, fear, anxiety, rage, sensory, pain, startling, we have the reticular formation, which is a grouping of white matter between the hypothalamus, the thalamus, cerebral cortex, cerebellum, and spinal cord. And specifically in the lecture, we were told to focus on the hypothalamus. And so the hypothalamus, which is located right here, is going to influence breathing in 3 main ways. The first is by changing body temperature through the hypothalamic thermoregulatory center. And then the second is, powerful emotions, fear, anxiety, rage will cause an increase in breathing. Right? If your body temperature is too hot, you can breathe more to cool off, right? We see this in panting in dogs a lot, right? But same mechanism works for people. Then we have the release of hormones. In particular, the hypothalamus is going to release thyrotropin-releasing hormone, and this is going to increase the expression of T4 and T3, as well as gonadotropin-releasing hormone, which increases progesterone. But the thyrotropin-releasing hormone, resulting in an overall increase in T3, T4, and progesterone, is going to stimulate breathing. All right, so the hypothalamus, as part of this reticular formation, grouping of interconnectedness of the white matter, aka the axons, is going to serve to stimulate the motor neurons. We know phrenic nerve for diaphragm, intercostal nerves for our friend, the external intercostal muscles, to help with breathing in. All right, and then they also synapse with the medulla, but just like with our voluntary motor control, it can bypass the motor control center, aka the medulla, and go straight to our motor neurons, which is what we see here. We have some bypassing of our control center. All right, so then that brings us to, we have our motor control center, but what's going on with our afferent side, our sensory side? We're going to be looking at the peripheral chemoreceptors, and then the central chemoreceptors, which are located on our medulla, and that's going to feed into our control center in the medulla, and then we know that will go out to affect our rate of breathing overall. All right, so let's get to it, starting by talking about our inputs, the peripheral chemoreceptors. And so the idea is that we have chemical chemoreceptors in the carotid body and in the wall of the aortic arch. So right here in the wall of the aortic arch, and this is going to synapse with the vagus nerve, aka cranial nerve 10. Then we have the carotid bodies at the bifurcation between the external carotid artery and internal carotid artery, and that's going to synapse with the cranial nerve 9, which is the glossopharyngeal nerve. Okay, so the locations of these peripheral chemoreceptors are here in the bifurcation of the common carotid arteries, and then here in the wall of the aortic arch. Importantly, we're not as focused on the aortic bodies, so these guys aren't going to have a huge impact on respiratory rate, whereas in humans, that's not the case in, say, other animals. For the carotid bodies, though, these guys right here, they are going to have a big impact on the rate of breathing. All right, so basically the idea is that these periphery chemoreceptors, though, are going to respond to changes in partial pressure of oxygen, in particular due to high altitude or, say, pulmonary disease. And they will, if you have an increase in partial pressure of oxygen, this is going to silence the carotid body's sort of tonic level, basic level of signal to the medulla. And if you have a decrease in oxygen, this is going to increase the activity of the medulla and therefore increase the rate of respiration right through the diaphragm, contracting. But this occurs for oxygen when you get to about 60 to 70 milligrams of mercury for the partial pressure of oxygen in the blood. And so that's what this diagram is trying to say. It says that, as you go this way, decreasing in the partial pressure of oxygen, once you get past this threshold of around 60 millimeters of mercury, the carotid body chemoreceptors, peripheral chemoreceptors, are going to say, hey, medulla, you need to do something about this. And so the medulla is going to cause an increase in ventilation overall. All right, it's important to know, though, that these cells, in particular, aka we said carotid bodies, but it's the glomus cells in the carotid bodies, are doing the partial pressure of oxygen sensing, but they also respond to an increase in the partial pressure of carbon dioxide. And so the carbon dioxide, we know if you have an increase in carbon dioxide, you're going to get an increase in the concentration of hydrogen ions. So basically, a decrease in oxygen or an increase in carbon dioxide will stimulate the medulla via the glossopharyngeal nerve, aka cranial nerve 9, to increase the rate of respiration. All right, and so what would cause a decrease in our oxygen content? For instance, high altitude, not breathing enough, some sort of diffusion defect, aka fibrosis. It's harder for oxygen to get into the blood. Some sort of mismatch of ventilation and perfusion, which we talked about in the previous set of lectures, and then a abnormal shunt of, aka mixing of poorly oxygenated blood with well-oxygenated blood. All right, but all of those would decrease your partial pressure of oxygen, and therefore all would stimulate the glomerulus cells inside of the carotid bodies located at the bifurcation of the common carotid arteries, and that would stimulate our medulla, which would stimulate our diaphragm to increase breathing. All right, so then we have our basically summary of what we just said. If you have a decrease in partial pressure of oxygen or an increase in carbon dioxide, you're going to have a net result of a decrease in arterial partial pressure of oxygen, an increase in partial pressure of carbon dioxide. Your peripheral chemoreceptor is going to say, hey, something's wrong. That's going to stimulate your medulla to say, hey, I need to breathe more. That's going to cause the diaphragm to contract. That's diaphragm contracting, increases the volume of the thoracic cage, which causes the lungs to expand, which causes inhalation, and therefore we can say it causes more ventilation. And the idea is that will get our bring our partial pressure of oxygen back up and decrease the partial pressure of carbon dioxide. All right, so this isn't anything that's particularly conceptually difficult. It's just knowing we have these peripheral chemoreceptors that can cause this reflex of getting you to breathe more if they're stimulated by lack of oxygen or too much carbon dioxide. All right, then we also have central chemoreceptors, and we're given this lovely diagram right here. And I say lovely somewhat ironically because basically this is just showing medulla, and the idea is that these central chemoreceptors are just located on the surface of this guy, right? And so we have a chemoreceptive sensitive area. And these chemoreceptors located on the surface of the medulla, they're going to be separated from the circulatory system by the blood-brain barrier, which is right here. And this has some effect because we have, therefore, less proteins. And so if you have, proteins are negatively charged, if you have an increase in carbon dioxide, then the carbon dioxide is going to diffuse into our cerebral spinal fluid past the blood-brain barrier. That will undergo a chemical reaction to form this bicarbonate ion and hydrogen ion. And so these central chemoreceptors are going to respond to an increase in hydrogen ion. And typically, you could buffer this increase in hydrogen ion just by having a whole bunch of negative proteins. And so in our cerebral spinal fluid, because we have this blood-brain barrier, we have less negative proteins overall to buffer against this increase in hydrogen ion concentration, which comes as carbon dioxide diffuses through the blood-brain barrier. All right, and so basically the premise is you have too much carbon dioxide, your central chemoreceptors on the medulla itself are going to be stimulated. If they're stimulated, they're going to stimulate the portion of the medulla that's going to then stimulate our respiratory muscles to contract, and you're going to try and get our carbon dioxide levels back to normal. All right, so that's what this diagram is just saying. It says, you have too much carbon dioxide in your arteries. That's going to increase the carbon dioxide in your extracellular fluid around your brain, which our cerebral spinal fluid, and then that increases the concentration of hydrogen ions. That's going to trigger the central chemoreceptors, these hydrogen ions. That's going to then trigger the medulla inspiratory center, which is going to send signals down the phrenic nerve and down the intercostal nerves to cause basically more breathing and get our remove the carbon dioxide from as much carbon dioxide from the blood as needed to get to a normal partial pressure of carbon dioxide. All right, so then now we have gone over kind of the central chemoreceptors and peripheral chemoreceptors, and in lecture we were given this truly awful graph, and I wrote on it with the hope that maybe this could be helpful, but I think that basically what we're trying to do is tie in the idea that these chemoreceptors are going to feed into the medulla via cranial nerve 9, the glossopharyngeal nerve, and cranial nerve 10, the vagus nerve, and they're going to the glossopharyngeal nerve, because we're saying that the aortic arch chemoreceptors aren't going to be doing very much here. The glossopharyngeal nerve is the one that's really important here, cranial nerve 9. The vagus nerve is going to come into play when we talk a little bit about the stretch and irritation receptors. We haven't gone over that yet. All right, but this graph over here is ventilation versus the partial pressure of carbon dioxide in our arteries. And so this first line right here is meant to be the efferent arm of the respiratory control loop. And so the idea is as you go to increasing partial pressure of carbon dioxide, then that occurs as ventilation decreases. So the idea is as ventilation goes down, then carbon dioxide is going to increase, which just intuitively, this entire graph is kind of what you understand intuitively. And then as ventilation increases, as we go this way, then the partial pressure of carbon dioxide is going to decrease. All right, and so then we have basically the idea of these chemoreceptor reflexes, and so the chemoreceptors are represented by these lines overall. And so basically what they're trying to say is that there is kind of a threshold value. So we have our normal equilibrium resting point for ventilation and the partial pressure of carbon dioxide. And if you get past this value, then you're going to trigger our chemoreceptors. And so once you get past a threshold value for our chemoreceptors, then you're going to have our ventilation increase linearly overall. And so that's what these straight lines are for. And you can see that, depending on the severity, right, if this is normal, then how far you are past out of normal. So these are showing us partial pressure of oxygen. If your partial pressure of oxygen is really low, then you get a steeper and steeper curve to increase the rate of rise of ventilation. All right, so maybe that's a little bit as clear as mud, but there's an important thing to also kind of take away from this graph, which is we do have something called the wakefulness drive. So if you're awake, you will never get to a ventilation of 0. That same is not true if you are asleep. All right, and so wakefulness drive will keep you at a minimum ventilation, but if you're asleep, you can have apnea occur, which is a stop of sleeping, or stop of breathing during sleep itself. All right, and so that's what this next topic is, which is during sleep, you don't have this sort of wakefulness drive that's maintaining a minimum ventilation. During sleep, you have basically stable breathing is controlled entirely by our peripheral and central chemoreceptors. And so we know that our peripheral chemoreceptors are, O2 and CO2 dependent, but our central chemoreceptors are, just CO2 dependent. So, really what we're focused on is the partial pressure of carbon dioxide beyond some sort of threshold that's going to cause us to breathe, ventilate. Right, and so the measures that determine stable breathing, are based on carbon dioxide and oxygen partial pressures, but snoring intensity. If you're snoring, you're not getting oxygen as well. Movements of the abdomen and rib cage, right? That should be like kind of consistent in the sense that it should be in and out for just part of breathing. Mouth, nose, oronasal airflow, and then measure of your oxygen saturation. So this is just a graph of basically these 4 different measures to determine if you have stable sleep, but our stable sleep is going to come as a result of our peripheral and central chemoreceptors measuring the carbon dioxide and O2. All right, so that's what these guys are. You can see, this is in a stable sleep. Our snoring is kind of kept to a minimum. It's not abnormal. Flow through the nose and mouth is consistent. Rib cage and abdomen are moving consistently. And then here, our O2 concentration is about 95%. All right, and then what if you have, how do you get unstable breathing? It's basically if you're below the chemoreceptor threshold, the chemoreflex threshold. And so this will, if you're below the chemoreceptor threshold, meaning you are below a certain amount of carbon dioxide, then your chemoreceptors aren't going to be stimulated, and you can have a temporary stop in breathing, apnea. You can have a decrease in, and as a result, a decrease in partial pressure of oxygen. And if you're not breathing, then an increase in carbon dioxide. And so that's what this graph is meant to show. I kind of found this graph to be a little bit unnecessary, but basically we have a curve for sleep apnea when you stop breathing, and then normal sleep. And the idea is that here we have ventilation versus partial pressure of carbon dioxide. If you get below a certain threshold for the partial pressure of carbon dioxide, then your chemoreceptors are going to stop being stimulated, right? And so you will have basically no drive to continue breathing because our carbon dioxide, if it's elevated, it says, oh, we have a lot of waste, we need to get rid of the waste, you need to breathe. Our chemoreceptors are saying to the medulla, you need to breathe. But if you don't have any carbon dioxide, then you are going to stop breathing. So that's when you get apnea. And then if you stop breathing, then you're going to have an increase in carbon dioxide and a decrease in oxygen. And then this increase in carbon dioxide is going to cause arousal and then hyperventilation to get back to normal. And unfortunately, for a lot of individuals, as we'll see on the next page, this arousal is not very significant, such that you can just fall back to sleep. And then as a result of having hyperventilated a little bit, now your carbon dioxide is really low again. And then if you're below this sort of threshold for your carbon dioxide, then you can stop your chemoreceptor stimulating the medulla, and then you get apnea again, and so you get this sort of like continuous cycle overall of carbon dioxide is low from hyperventilation, then you stop breathing, then carbon dioxide goes up, you wake up, and it's not good. So this is basically just kind of describe what this entire thing was saying, but if we're dealing with the cycle of not breathing, so here we have somebody with apnea, you can see that the flow of breathing is stopping right here, and then as the flow of breathing is stopping, you have a buildup in carbon dioxide, which you can see here, it's starting to increase, and then you have a buildup in carbon dioxide, then you have a triggering of, you just start to get a triggering of the inspiratory muscles, and then you have movement of these muscles, increase in oxygen flow, you're hyperventilating, then if you're hyperventilating, then carbon dioxide is going to go down, and you stop breathing, and then your carbon dioxide builds back up again, you have your rib cages get activated, and so you have this cycle overall. And so that's what the cycle is saying. Stop breathing, you have reduced stimulation of the hypoglossal nerve, which is basically keeping your airway diameter open. And if you have a decrease in airway diameter, airways collapsing, you're going to have a stop in breathing, increase in carbon dioxide, decrease in O2. Then you're going to have arousal from deep sleep because now your chemoreceptors are in fact being stimulated. Your airway is going to open as this hypoglossal nerve is going to be stimulated. Then you breathe too much to correct it. And then if it's overdone, then you have low CO2 or receptors aren't getting stimulated. Then you repeat the cycle over again, which is happening to this poor individual up here. All right, and so then right here, this is just another diagram from our lecture, which is just saying, below a carbon dioxide threshold, you don't have spontaneous breathing, as our breathing during sleep is reliant on our central and peripheral chemoreceptors. And so for this poor individual, they had intentionally hyperventilated him to reduce the carbon dioxide, so you have low carbon dioxide. And you can see that as you have low carbon dioxide, so you go from left to right, our airway is decreasing in size until he kind of wakes back up as our carbon dioxide starts to increase again. And so the idea is that after hyperventilating, you have low CO2, your central and peripheral chemoreceptors not being stimulated. That results in decrease in stimulation of the medulla, and therefore decrease in stimulation for this particular muscle, which is keeping our airway open, called the genioglossus muscle, which is due to reduced hypoglossal nerve stimulation, which is from stimulation from the medulla, and then so our airway decreases. But overall, our breathing is a result of these peripheral and central chemoreceptors, our carotid body chemoreceptors going up to our medulla, and then for the airway, the medulla innervating the gloss, stimulating the hypoglossal nerve, which stimulates this genioglossus muscle, which keeps the airway open. All right, so then we're almost done. Other neuronal inputs. If we go back to let's see, this diagram right here, we said we had, our peripheral chemoreceptors, the ones in the walls of the aortic arch, really aren't doing too much in regulating respiratory rate for people, but we do have that vagus nerve, which would innervate the, take sensory data from the aortic arch, is also going to give us some sensory data from receptors inside of the airway and the lungs themselves. And so what are those receptors? Those are our pulmonary stretch receptors and then our irritant receptors. So basically the idea is that the lungs and the conducting airways have receptors and sensors as well, right? We've talked so far about receptors of the bifurcation of the common carotid arteries, so they're sensing blood flow and partial pressures in the blood. But these guys are just stretch receptors and, is there something here receptors, aka irritant receptors, so not oxygen or carbon dioxide sensing. These are in the lung connective tissue for the pulmonary stretch sensors. They respond to stretching due to rapid and sustained inflation of the lung. They're connected to the vagus nerve, which goes up to the medulla, and they will basically stimulated by pulmonary stretch to inhibit the DRG, which is the dorsal respiratory group in the medulla, which is going to say stop breathing in and start breathing out. Right? And so this reflex of these pulmonary stretch sensors saying, hey, the lung is really stretched and it's stretched for a long time. This is rapid and sustained inflation. We're going to trigger the medulla via the vagus nerve, inhibit the dorsal respiratory groups in the medulla, start to breathe out and stop breathing in. And this reflex is called the Herring-Brewer reflex. Right, and so we have our pulmonary stretch sensors inside of the lungs themselves. It's important to note if the vagus nerve is cut and you don't have this reflex, and breathing in will not be inhibited, it will be prolonged. Right, generally this reflex occurs when the tidal volume, your normal breathing in and out, is 3 times the normal volume. All right, then we have our irritant receptors. Basically, this is responding to chemical and mechanical stimuli. These promote protective reflexes. They're found in the airway from the nose to alveoli. And so, you think of it as you have something in your nose, you're going to sneeze. And so all of these receptors are going to cause an increase in breathing and bronchoconstriction. So in the nose, you have the nasal receptors. You have the epipharyngeal receptors for, if you accidentally breathe in food or water into the conducting part of the respiratory system, then you're familiar with coughing quickly. Laryngeal receptors for coughing, tracheal receptors for coughing via the vagus nerve. Then you have these J receptors in the alveoli themselves, and so they will respond to engorgement, enlargement of the pulmonary capillaries, and increased interstitial fluids. The J receptors synapse with the vagus nerve, and they're going to cause rapid shallow breathing, and they can also cause apnea. All right, and so our irritant receptors are dealing with particles. Generally, small particles get trapped by mucus, swept up by cilia, or eaten by macrophages and then pooped into the lymphatic system. Large particles are going to get trapped generally in the mucus of the nose or the pharynx overall. Right, so we have our nasal cavity, we have our pharynx, and these irritant receptors are really throughout the entire respiratory system overall. All right, so that's it. We do have some review questions for this particular lecture, and they're in the review document, so I encourage you to look at them. And that's it. Just a quick reminder, we have our singular equation for O2 content in blood, milliliters of oxygen per 100 milliliters of blood overall. All right, we'll see you next time. Bye, guys. Good job.